Hi, I'm Aurel Enriquez, and this video contains our discussion on Chapter 19 of Mahon's textbook of Diagnostic Microbiology, which focuses on the order Enterobacterales. Here is the outline of our discussion. As usual, we'll start off with the general characteristics applicable to every member of Enterobacterales, and then we'll go on to discuss the opportunistic Enterobacteriaceae species. We'll be looking further onto the different strains of Escherichia coli before moving forward to the other clinically significant opportunistic species, as we can see right here on this second column. After that, we'll look into the true pathogens from the family. And lastly, alamin po natin kung alin-alin yung mga culture media commonly used in the isolation of Enterobacterales members. Let's proceed with the general characteristics. Enterobacterales members are facultative anaerobes, which, in case you forgot, it means that they can grow either in the presence or absence of oxygen, but they will grow better if oxygen is present. So again, facultative anaerobes po ang Enterobacterales members. They are also non-spore forming gram-negative bacilli. All members of Enterobacterales are glucose fermenters. So, if the organism was not able to ferment any of the sugars present in the TSI, then this is most likely not a member of Enterobacterales. All enterobacteria have peritrichous flagella and are motile at 35 degrees Celsius, except for three members, yung SKY or SKY, Shigella, Klebsiella, and Yersinia. So Klebsiella is one of the opportunistic pathogens. Meanwhile, yung Shigella and Yersinia, they are the true pathogens. So later, as we go along through our discussion, i-explain ko po what we mean when we say true pathogens or primary intestinal pathogens versus opportunistic Enterobacterales. Next, all Enterobacterales are non-encapsulated except for two, yung EK. Okay, EK. So, EK stands for Enterobacter and Klebsiella. All right, so since these two are encapsulated, we could expect that in culture, they would produce mucoid colonies. Okay, so ulitin po natin, all members of Enterobacterales are motile at 35 degrees Celsius except for SKY or Shigella, Klebsiella, and Yersinia. Next, all members are non-encapsulated except for two, which are EK, Enterobacter and Klebsiella. Now, after gram staining and under the microscope, all members of Enterobacterales can be seen as straight gram-negative rods with rounded ends, as we can see right here on these photos. So ito po, right here, and these ones right here. Okay? So as we proceed with the discussion, hindi ko na po ipapakita ulit yung microscopic morphology nila individually because they all look like this. Okay? In the past, all of the genera that we will be discussing in this chapter belonged to one family, which is Enterobacteriaceae. However, classification of the members had changed. So new species have been discovered, and some species have been moved to a different taxonomic classification. So this slide shows us all the bacterial genera and species that are currently classified under the order Enterobacterales and under the family Enterobacteriaceae. Shown here are all the other members of the order Enterobacterales that have been reclassified into different families because, again, in the past, they were all under Enterobacteriaceae. Okay, ulitin po natin, no? And maybe you might be able to find in older editions of our reference textbook na ang title po ng chapter na ito noon ay Enterobacteriaceae. Pero ayun nga po, no, may mga bacterial genera na na-reclassify into a different family. So instead of having just one family, which is Enterobacteriaceae, meron na siyang isang order na Enterobacterales, tapos there are multiple different families. So as we can see right here, maliban sa Enterobacteriaceae, nakikita po natin na merong Morganelliaceae, Yersiniaceae, and other families like that. So again, yung mga genera na nakikita po natin dito, again, in the past, they all belong to the family Enterobacteriaceae. But again, they have been reclassified into these different families, but they are still under the order Enterobacterales. Let's now talk about antigen determinants for serological classification. There are three antigenic determinants that can be used for the serologic identification of infections that may be caused by Enterobacterales. And these are antigens that can be found on different parts of the bacterial cell. So one of these is the somatic antigen, which can be found on the cell wall. Okay, ulitin po natin. The somatic antigen is located specifically on the cell wall. So the somatic antigen is otherwise known as the O antigen, which is heat stable. All right, heat stable siya, which would set it apart from the other two antigens kasi po itong dalawa, they are heat labile. All right, meaning that they can be destroyed if the sample is subjected to heating. Okay, but again, the somatic antigen is heat stable, so its structure will not be affected even after heating the organism. So in some serologic methods, if they want to increase the specificity of the test in detecting the O antigen, they could subject the sample to heating, which eliminates the interference of the two other antigens. And there you have it. Yung somatic O antigen lang po yung madedetect natin. Note that the somatic O antigen is used in the serotyping of Escherichia coli and Shigella species. Next is the flagellar antigen, also referred to as the H antigen. So as I've already mentioned, this is heat labile and it can be found on the flagella of the bacterial cell, as the name implies. Okay, so again, what type of flagella do Enterobacterales members have? And they are peritrachous. Okay, so the H antigen is used for the serotyping of Salmonella. Third is the K antigen or the capsular antigen. Now, this is also heat labile and for different species, may kanya-kanya din pong pangalan ang K antigen. So for Escherichia coli, the K antigen is referred to as the K1 antigen. Para naman po sa Salmonella enterica, its K antigen is called the V or VI antigen. So please don't forget, no, here is an illustration pointing out where these antigens can be found specifically on the bacterial cell. All right, so for visual learners, no, this one is for you. Again, ang somatic antigen or O antigen, nasa cell wall ito makikita. The capsular antigen or the K antigen is found at the capsular structure and the flagellar H antigen is at the flagella, as we can see right here in this diagram. So again, these are the different antigenic determinants for serological classification of some Enterobacterales members. Let's now discuss the opportunistic members of Enterobacterales, including their associated infections, as well as their colony morphology and some biochemical test results. So, when we say opportunistic members, all of these species are actually part of the normal flora of humans. And as with all other opportunistic organisms, these do not generally cause disease in healthy, immunocompetent human hosts. Okay? Do take note, though, that they still have some significant virulence factors and may still cause extra intestinal infections outside of their normal body sites. Escherichia coli, commonly known as the colon bacillus, is the most clinically significant species in the genus. May mga iba pa pong species na kasama sa genus na to, such as Escherichia hermanii, Escherichia vulneris, and Escherichia albertii. But again, focus muna po tayo sa Escherichia coli dahil dito pa lang ang dami na nating kailangan aralin. Okay, so in water testing, or the bacterial analysis of water purity, which is usually done for water supply companies and water refilling stations, malalaman po natin na possibly contaminated with fecal material yung water supply if we are able to isolate Escherichia coli from the sample. Okay? So yun po yung ibig sabihin ng statement na to. Now, Escherichia coli is the primary indicator of fecal contamination in water purification. So ulitin po natin, from a water supply, if we were able to isolate Escherichia coli, this might mean that the water supply is contaminated with fecal matter. All right? Clinically speaking naman po, Escherichia coli has been documented as the leading cause of nosocomial urinary tract infections. Okay, so this bacteria has both types of fimbriae or pili, and these would serve as its virulence factor. Okay, so meron sila both nung conjugation pili and yung common pili or the adhesive fimbriae. So the common pili, it helps the bacteria to attach to surfaces. Other examples of bacteria that have the common pili are Neisseria gonorrhoeae and the Pseudomonas species. The other type of pili is the conjugation pili or sex pili, which is used by the bacteria during the process of genetic transfer, specifically conjugation. So recall lang po, no, recall, during conjugation, one bacterial cell can transfer or donate some of its genetic material to a recipient cell. So ang problema po, if the donor cell contains a DNA that carries the genetic code for antimicrobial resistance, pwede niya po itong ipasa to other cells that do not yet have the code for antimicrobial resistance. So isa po ito sa mga ways on how multi-drug resistant organisms are spread. So as previously discussed in your laboratory classes, specifically on chapter 9, alam nyo na po yung meaning nung INVIC, diba? So let's look at this one right here. So, indole production positive, methyl red positive, Voges Prescour negative, and citrate negative po ang INVIC reaction ng Escherichia coli. So its INVIC is positive positive, negative negative. All right? So ito naman po yung TSI results niya. It is A over A or acid over acid, positive for gas production and negative for hydrogen sulfide reaction. So after we take a look at the colony morphology of Escherichia coli on different cultural media, isa-isahin din po natin yung mga strains ng Escherichia coli on the following slides. On blood agar plates, most strains of Escherichia coli are non-hemolytic and they look like this first photo right here. Some strains have been described as beta hemolytic, but generally non-hemolytic po ang karamihan ng Escherichia coli. On MAC or the McConkey agar, they are seen as flat, dry, and pink-colored colonies with a pink discoloration on the agar on the immediate surrounding of their growth, which tells us that they are lactose fermenters. Okay, so yun po yung meaning ng LF, lactose fermenting. Although there are a few strains that have also shown to be non-lactose fermenters. Okay, ulitin po natin, may mga strains daw po ng Escherichia coli that are non-lactose fermenters. But again, majority po ng Escherichia coli strains are LF or lactose fermenting. On EMB or the eosin methylene blue agar, which we'll talk more about later, dito po natin makikita yung characteristic green metallic sheen colonies ng Escherichia coli. So Escherichia coli produces a green metallic sheen on the Eosin methylene blue agar because of its intense lactose fermenting capabilities, creating high concentrations of acid that causes the Eosin and methylene blue dyes to precipitate, forming a metallic luster. Okay, so yun po yung dahilan kung bakit may green metallic sheen ang Escherichia coli on EMB. Okay, ulitin po natin, it's because of the intense lactose fermenting capabilities of Escherichia coli, leading to the precipitation of eosin and methylene blue dyes, which we would not be able to observe among other bacterial organisms. So kapag nakita po kayo ng green metallic sheen on EMB, dapat ang una nyo na po agad maisip ay Escherichia coli. Because again, this colony morphology is unique for E. coli. Meron pa pong just a side kind of like side info, no? Meron pa pong isa pang non-fermenting gram-negative bacilli that has also been described to produce a metallic sheen. Pero ito po ay sa BAP naman natin ma-observe. And that is Pseudomonas aeruginosa, as shown here in this pop-up picture at the top right. So, it's quite nice to look at, no? Maganda po sila tignan, no? So, ayan, again, on BAP, no? Pag may metallic colonies po kayo na nakita on blood agar plate, it's most likely Pseudomonas aeruginosa. Pero kapag sinabing EMB, tapos green metal sheen po ang na-observe natin, then what we're looking at is Escherichia coli. Alright? Next is XLD or the xyloslicine deoxycholate agar na madidiscuss din po natin mamaya. So E. coli will produce large, flat, yellow colonies, as you can see right here on this fourth picture. Let us now discuss the different pathogenic strains of E. coli. First up is the uropathogenic E. coli or UPEC. This is the most common cause of urinary tract infections in humans. So UPEC possesses the common pili. All right, so it's not used for gene transfer. Hindi katulad no conjugation pili, but rather the common pili adheres strongly to the urinary tract of the host, making it very difficult to wash out simply by urination. Okay, so if you would notice, no, if you or anyone you know has ever had urinary tract infections or UTI, mapapansin po natin na isa sa mga clinical manifestations ng urinary tract infections is frequent urination, 'di ba? But again, that's not enough to resolve the infection, 'di ba? Hindi po 'yun sapat to wash away the bacteria that's causing the infection. And again, it's because of their adhesive fimbriae or yung common pili nila. Because again, the common pili is used for adherence, meaning magiging makapit sila dun sa mucosal surfaces that they're currently infecting. That is enough, no? They adhere enough that they don't get easily washed out by urine flow. All right. So again, that's one of the virulence factors of the uropathogenic E. coli. Next is the enterotoxigenic E. coli or ETEC strain. So this has been associated with what has been known as the traveler's diarrhea. Okay, again, the ETEC strain is known to cause the traveler's diarrhea. So nabanggit po natin kanina, di ba, that E. coli is a primary indicator of fecal contamination of water supplies, right? Now, the reason why it was referred to as traveler's diarrhea is because when people travel to, let's say, for example, third world countries or regions that are very poor and hindi pa po ganun ka-develop yung mga water purification systems nila, their water supplies or water sources are most likely contaminated by many enteric pathogens. So, kapag po itong isang traveler na to, for example, nakainom siya ng tubig galing sa lugar na ito, this individual will now develop diarrhea and itong ETEC po, yung strain, that was predominantly isolated from those cases of diarrhea. So kaya po naging ganon yung tawag sa disease, the traveler's diarrhea. The preferred habitat of ETEC in the human body is at the proximal region of the small intestine. Also, take note that people who are suffering from achlorhydria are the population who are at most risk for acquiring the infection from the ETEC strain. So, what is achlorhydria? This is the deficiency of hydrochloric acid production in the stomach. All right, so maliban po kasi sa digestion, the hydrochloric acid in the stomach also helps us eliminate some pathogenic organisms, di ba po? Because some of them cannot survive the extreme level of acidity. So ngayon, kung kulang po ang stomach acids in the case of people with achlorhydria, that is one less first line of defense for the host to fight off both parasitic and bacterial pathogens. Now, the virulence factor of the ETEC strain comes from the production of both heat stable and heat labile enterotoxins. Next is the entero-invasive E. coli or EIEC. This produces a dysentery-like or Shigella-like infection, which we'll get to know more about later once we talk about the Shigella species. Now, the preferred habitat within the human host is within the intestinal epithelial cells. Kaya po ito tinawag na entero-invasive because it can penetrate or invade intestinal cells. Next is the EPIC strain or enteropathogenic strain, which is the causative agent of infantile diarrhea. That doesn't mean that it exclusively causes diarrhea among infants. It just means that most cases involve infants or children under 2 years old and below. All right. So the EPIC strain does not produce any toxins, but it can cause cell damage, which leads to the loss of microvilli, which is termed as effacement. All right. Ulitin po natin, the loss of microvilli or destruction to the structure of the microvilli is termed as effacement, as we can see right here in this illustration. Now, this damage to the structure of the microvilli would in turn affect its function, which is absorption. So, this may temporarily hinder the capability of the gastrointestinal tract to absorb nutrients. So, again, that's for the enteropathogenic E. coli or the EPIC strain. Next is the enterohemorrhagic E. coli or EHEC, specifically the EHEC serotype O157H7. So ano po yung meaning ng mga letters na ito? Ito po yung mga serologic antigens na discuss natin kanina, di ba? The somatic O antigen 157 and the flagellar H antigen number 7. So the virulence factors of the EHEC strain would include Shiga toxins 1 and 2, also known as virotoxins because of their capability to destroy viro cell lines from African green monkey kidneys. So these toxins have the capability to destroy vascular endothelial cells, which is why these are the types of diseases that it can cause: hemorrhagic colitis, bloody diarrhea, and HUS or the hemolytic uremic syndrome. So ano ba ang hemolytic uremic syndrome? HUS is a condition in which the small blood vessels of the kidneys are damaged and inflamed. All right, so that is for the enterohemorrhagic E. coli O157H7. Now, this one is a subset of the STEC or the Shiga toxin producing Escherichia coli. Kaya po, ayun no, nabanggit natin kanina, its virulence factors are the Shiga toxin 1 and Shiga toxin 2. Again, that is the EHEC serotype O157H7. Last strains are the enteroadherent and the diffusely adherent E. coli or the EAEC and DAEC. So in a lot of references, they're also referred to as the enteroaggregative E. coli. All right, so ulitin po natin, these strains are referred to as either the enteroaggregative, enteroadherent, or diffusely adherent E. coli. So those are three different terminologies, but we are talking about the same strain. All right. So they have been documented to cause both urinary tract infections and watery diarrhea. Okay. So kanina po, di ba, we have the UPEC strain or the Uropathogenic E. coli. So the Uropathogenic E. coli, they're only documented to cause UTI. Okay. Pero if it's able to cause both UTI and watery diarrhea, then yung EAEC na po ang pinag-uusapan natin. So they are called enteroaggregative because they have been observed to stick together in the intestinal mucosa, as we can see right here in this illustration on the left, and this electron micrograph on the right. Okay? So ayan po, no, they tend to aggregate together, giving them a stacked brick appearance. So tingnan po natin itong illustration na to and itong electron micrograph na to. Kapag nagsama-sama daw po sila, because of the microscopic morphology of Escherichia coli, again, ano po ulit yung general microscopic morphology ng Enterobacterales members? They are gram-negative bacilli. So they're gram-negative bacilli that are straight with rounded ends. Okay, so medyo mukha nga silang bricks. All right, so again, the enteroaggregative E. coli has been described to have a stacked brick appearance because they tend to aggregate or stack upon each other, as we can see right here in this illustration and in this electron micrograph. All right, so ulitin natin, kapag sinabing stacked brick appearance and it's capable of causing both UTI and watery diarrhea, dapat ang maisip nyo na agad is the enteroaggregative E. coli. Let's now just quickly go through some of the other Escherichia species, which is only occasionally capable of causing infections in humans. First is Escherichia fergusonii. So Escherichia fergusonii has been isolated from urine samples, blood, wound infections, feces, and gallbladder. And next is Escherichia albertii. So Escherichia albertii has been observed to carry two different genes. All right. First is the STX2F gene, which could code for Shigella-like toxin production. All right. Ulitin po natin, if this specific strain of Escherichia albertii carries the STX2F gene, then it may be able to produce a Shigella-like toxin. Next, it could also carry the TB gene, which codes for the toxin found in Clostridioides difficile. All right, so ulitin po natin, if Escherichia albertii carries the TB gene, then it may be able to produce the same toxin that could be found in Clostridioides difficile. So they're actually quite easy to remember. No, it may look confusing at first kasi parang what are these random letters and numbers, diba? But if you look at the first two letters, no, ST, then it could code for the Shigella-like toxin, diba? Next, itong CDTB, ang first two letters ay C and D, which could lead to the production of the same toxin found in Clostridioides difficile. Alright? So, ayun po. How do we remember kung ano yung type of toxin na pinoproduce ng genetic material or genetic code na ito? It's the first two letters. Again, the STX2F gene codes for a Shigella-like toxin. Meanwhile, the CDTB gene codes for the same toxin found in Clostridioides difficile. Next organism na po tayo, Klebsiella pneumoniae, subspecies pneumoniae. Grabe no, parang slide 16, nasa slide 16 na tayo. Pero mula dun sa slide 1 hanggang slide 16, halos puro Escherichia coli lang yung napag-usapan natin. So marami po talagang iba't ibang organisms tayong mapag-uusapan dito sa Enterobacterales. But anyway, let's now proceed to the next one, which is Klebsiella pneumoniae, subspecies pneumoniae. The common name for this one is the Friedlander's bacillus. This is the most commonly isolated species of Klebsiella. So of course, meron pa pong iba katulad ng Klebsiella pneumoniae subspecies ozone or Klebsiella oxytoca or Klebsiella rhinoscleromatis. But as I've said, now my discussions will be focusing on the most significant isolates. Okay, so Klebsiella pneumoniae subspecies pneumoniae is the causative agent of community-acquired pneumonia, which is characterized by expectorating a current jelly-like sputum. So currents are a type of like small berries, as we can see right here in this photo. All right, these are currents. Okay. Now, so sinasabi po nila na yung sputum daw po nung patient ay kahawig nung jelly or jam that is made out of these berries. All right, which is quite a unique description. No? It's not a very common thing here in the Philippines. Sa Pilipinas po, kapag tayo ang nasunod sa pagpapangalan ng sputum na yan, tatawagin lang siyang ketchup-like sputum. It literally just looks like ketchup, no? Ang social pa pakinggan nung current jelly-like sputum. But anyway, ayan po, no? As I've mentioned earlier, in the general characteristics, Enterobacterales are non-encapsulated except for two, diba? Which would include Klebsiella. So, ito pong polysaccharide capsule na to, this serves as their virulence factor. Now, in relation to that, dahil nga po encapsulated sila, no, it will be very obvious in their colony morphology. So on McConkey, we can observe these colonies to be pink, lactose fermenting, mucoid colonies. All right? So kahit po dito sa BAP, no, they always appear very shiny, almost kind of like wet looking, and it is reported to be mucoid. All right. So again, that is the colony morphology of Klebsiella pneumoniae subspecies pneumoniae, or Klebsiella species in general. Again, they have a polysaccharide capsule, so we could expect that they have a mucoid appearance. All right. Now, since encapsulated nga po ang Klebsiella, this will also show a positive reaction if we were to subject them to the Kellang capsular swelling test. All right? So, saan po natin unang na-encounter ang Newfield Kellang capsular swelling test? During our discussion on Streptococcus pneumoniae, di ba po? So, kung nakalimutan nyo na yung principle and procedure ng Newfield-Kellang test, please take time to review our discussion on chapter 15. All right? But then again, if an organism is or it possesses a polysaccharide capsule, you could expect that it will test positive for the Newfield-Kellang reaction. Ano po yung biochemical test reactions ng Klebsiella pneumoniae? Klebsiella pneumoniae subspecies pneumoniae is indole negative, methyl red negative, which would automatically mean that it's Voges-Proskauer positive, di ba po? As I've said in my discussion on chapter 8, di ba? No clinically significant bacteria can be positive for both MR and VP. And lastly, it's citrate positive. So kung babasahin natin yung INVIC niya, its correct and complete INVIC reaction is negative, negative, positive, positive. Alright, ulitin po natin. And compare natin, no? For Escherichia coli, kanina, ano yung correct and complete INVIC ng Escherichia coli? It's positive, positive, negative, negative, diba? But for Klebsiella pneumoniae subspecies pneumoniae, baliktad. It's the other way around. So it's negative, negative, positive, positive. All right. So again, if you would notice, magkabaliktad po ang INVIC reaction ng Escherichia coli and ng Klebsiella pneumoniae. Another reaction that could set them apart is that Klebsiella pneumoniae subspecies pneumoniae is also urease positive. Alright? Pero urease negative po kasi ang Escherichia coli. Alright? Again, Klebsiella pneumoniae is urease positive. But if we look at their TSI reactions, dun sila magkaparehas. Alright? The TSI reaction for Escherichia coli and Klebsiella pneumoniae is the same. It's acid over acid, positive for gas production, and negative for H2S production. Okay? A over A, acid over acid, positive for gas, negative for H2S. Parehas ang E. coli and Klebsiella pneumoniae. Let's now talk about other Klebsiella species that have been associated with numerous infections. First is Klebsiella pneumoniae subspecies ozonae, which has been isolated from nasal secretions and cerebral abscesses. So this organism causes atrophic rhinitis, which is characterized by the formation of thick, dry crust on the nasal cavity, which has resulted from the progressive wasting away or decrease in size, termed as atrophy of the mucous nasal lining. Okay, so in general, Klebsiella pneumoniae subspecies ozonae has been linked to tissue destructive diseases restricted to the nose. Okay, so upon exploration of the nasal cavity, this is what the infected areas would look like, as we can see right here in this picture. So Klebsiella pneumoniae subspecies ozonae is highly associated with the presence of plasmid-mediated ESBLs, contributing to the large numbers of antimicrobial resistant hospital-acquired infections. So ESBLs, or microorganisms that produce enzymes known as extended spectrum beta-lactamases, are resistant to most beta-lactam antibiotics, including penicillins and cephalosporins. Next is Klebsiella pneumoniae, subspecies Rhinoscleromatis, which was formerly referred to as a separate species called Klebsiella rhinoscleromatis. So this has been isolated from patients with rhinoscleroma, which is an infection of the nasal cavity that manifests as an intense swelling and malformation of the entire face and neck. So this photo on the lower right shows us a case of a 63-year-old man that presented with a 10-year history of a mass growing in the central region of his face, involving his nose and upper lip. So histopathological examination revealed many foamy macrophages, and the culture of tissue fragments showed the presence of Klebsiella. Okay. Next is the Enterobacter species, specifically the members of the Enterobacter cloaceae complex. So in here, the most common isolates are Enterobacter cloaceae and Enterobacter hermaceae. So kaya po sila naka-highlight, no? These two are the most common isolates. In the past, we also had Enterobacter aerogenes, but this has been reclassified as Klebsiella aerogenes. They may resemble Klebsiella on Mac, and why is that? Bakit daw po magkahawig ang Klebsiella at Enterobacter on Maconchi agar? Because parehas po silang encapsulated, 'di ba? So I didn't include a picture anymore to make space for the this other content, no. But again, you would expect na pink, mucoid and lactose fermenting colonies din po ang Enterobacter species. So even in their INVIC and TSI reactions, magkaparehas po ang Klebsiella and Enterobacter. So the INVIC of Enterobacter species would be negative, negative, positive, positive. Its TSI reaction would be acid over acid, or A over A, positive for gas, negative for H2S. Okay, so kung mapapansin nyo po, ganyan palagi yung kumbaga way on how we report TSI reactions. So kahit yung negatives po, ilalagay pa rin natin. So dapat kompleto palagi yung ire-report nyo kapag TSI po ang binabasa natin. So you include the slant, the but, the gas production, and H2S production. So in this case, no, it's A over A, positive for gas, and negative for H2S production. So kahit negative po, i-include nyo pa rin yan dun sa complete reporting. Right? Cronobacter is also included in the family Enterobacteriaceae, which contains 7 different species, but the most common human isolates would include Cronobacter sakazakiae and Cronobacter malonaticus. So, Cronobacter sakazakiae has been documented as a pathogen in neonates. All right. So it could cause meningitis, bacteremia, or necrotizing enterocolitis. And this could be recognized by its production of yellow pigmented colonies. So on this photo, we're able to see, ayan po, no, on McConkey agar, this is a mixed colony of Enterobacter cloaceae and Cronobacter sakazakiae. So nakikita po natin those colonies that's capable of producing yellow pigments. Those are Cronobacter sakazakiae. So again, Cronobacter sakazakiae is a neonatal pathogen. Next is Cronobacter malonaticus. Now, this one is more commonly observed among adults. No? So they're biologic or biochemically, they're biochemically similar to one another. So it's very difficult to actually differentiate them in the laboratory, and it would require DNA sequencing para po ma-differentiate natin ang Zakazakii and Malonaticus. But again, with regards to their target demographic or population at risk, mas common na na-observe among neonates ang Cronobacter sakazakii and mas common na na-observe among adults ang Cronobacter malonaticus. And again, if we were to look at their colonies, pag yellow pigmented po, it is Cronobacter sakazakii. There are two notable isolates under the genus Citrobacter, the Citrobacter florentii complex and Citrobacter corseri. Citrobacter florentii is commonly recovered from diarrheal stool cultures. Now, about 80% of Citrobacter florentii complex isolates produce a positive hydrogen sulfide reaction. Meanwhile, some strains, about 50% of the isolates, fail to ferment lactose. Now, the colony morphology of Citrobacter florentia and primary selective media can be mistaken for Salmonella when isolated from stool cultures. Next is Citrobacter corseri, which causes a totally different clinical manifestation, such as neonatal meningitis and brain abscesses, as we can see right here in this CT scan. All right, so again, Citrobacter corseri has been known to cause brain abscesses. Pero ang Citrobacter florentii complex po, mas na-observe siya among diarrheal samples. Now, being late lactose fermenters, pwede pong magbago ang itsura ng colonies ng Citrobacter depending on how long you incubate your samples. So within 24 hours, they would appear as the typical clear lactose or clear, colorless, and non-lactose fermenting colonies. Kaya po, madalas silang mapagkamalan as salmonella dahil ang salmonella po, they are in fact non-lactose fermenting. But if we were to extend the incubation for up to 48 hours, makikita natin na nagbago na siya. Okay, nagbago na siya. Citrobacter colonies can be seen as light pink, late lactose fermenting colonies after 24 hours of incubation. All right, ulitin po natin. Within the usual 18 to 24-hour incubation, clear, non-lactose fermenting muna ang itsura ng Citrobacter species. But if you extend the incubation period for up to 48 hours, doon lang po sila magdi-display ng lactose fermentation. Alright? So that would take about 48 hours. Okay? So ayun po, no? Dito sa genus na to, kahit hindi ka mag-ONPG test, i-extend mo lang yung incubation period, malalaman mo kung late lactose fermenter siya. But if, for example, you don't have 48 hours, diba? You don't have that much time to extend the incubation for you to be able to observe late lactose fermentation, you could perform what is known as the ONPG test, all right? The orthonitrophenyl beta D galactopyranoside test. Now, this one is done to observe late lactose fermenters. So kung halimbawa, na-observe natin na upon 24 hours of incubation, non-lactose fermenting yung colonies, we could subject it to the ONPG test. Now, if it still produces a negative result on ONPG, then we could confirm na non-lactose fermenting talaga yung organism. But if, for example, upon 24 hours of incubation, non-lactose fermenting yung colonies, and then we perform the ONPG test. Kahit hindi na natin extend yung incubation period, if it tests positive for ONPG, then that indicates that the organism is a late lactose fermenter. Okay? Now, here are the other biochemical test results of Citrobacter forundii versus Citrobacter courseri. On IMVIC, isa lang po ang pinagkaiba nila, and that is indole production. Let's repeat. Citrobacter forundii complex, it produces a negative, positive reaction wherein the indole is negative. But for Citrobacter courseri, its indole reaction is positive. Next is Plesiomonas shigelloides. Plesiomonas was formerly under the family Vibrionaceae because of its phenotypic characteristics, but further studies showed that it is actually more closely related to Enterobacteriaceae. Now, two things that make it stand out from the rest of Enterobacterales members is that Plesiomonas shigelloides is the only oxidase positive member and it is a non-glucose fermenter. Okay, ulitin po natin. Enterobacterales members are mostly oxidase negative. Pero ang Plesiomonas shigelloides po, it is the only oxidase positive member. Next, kanina po nabanggit din natin sa general characteristics, Enterobacterales members are glucose fermenters, diba? But Plesiomonas shigelloides is negative for glucose fermentation. Also, one of its similarities to Vibrio is that it is also sensitive to the O129 Vibrio static agent. Now, Plesiomonas shigelloides is a straight gram-negative bacilli, which is not encapsulated and non-spore forming, and it's also motile. So all of those descriptions is similar with all the other Enterobacterales members. They may occur singly or in pairs or sometimes in short chains or filaments, all right? Now, they have a monotrichous flagella, or they could also have up to 5 lopotrichous flagella. Its antigens could cross-agglutinate with Shigella sonnei, Shigella dysenteriae, and Shigella boydii, which is why it's most probably why it was named Plesiomonas shigelloides, all right? Because it has some Shigella-like characteristics. Let's now look into the diseases associated with Plesiomonas shigelloides. Cases of gastroenteritis due to Plesiomonas shigelloides are usually self-limiting and is acquired most often after the consumption of undercooked seafood or untreated water. All right, ulitin po natin ang mode of transmission ng Plesiomonas shigelloides infections is via ingestion. So there are three major clinical types of gastroenteritis caused by Plesiomonas shigelloides. So one is the simple watery diarrhea. Two is the subacute or chronic disease that can persist from 14 days to as long as 3 months. So imagine having diarrhea for up to 3 months, right? Um, I'm sorry. Third is the most serious one, and it can be invasive and resemble colitis. All right. So, ano ba ang ibig sabihin ng colitis? Colitis is characterized by the inflammation of the inner lining of the colon or a segment of the large intestine. All right. So, ulitin po natin, there are three clinical types of gastroenteritis that is caused by Plesiomonas shigelloides. First is the regular watery diarrhea. Next is the subacute or chronic type, which could last for up to 3 months. Again, imagine yourself having diarrhea for up to 3 months. And the third type is the invasive dysenteric form, which again resembles colitis. Extraintestinal infections happen commonly to those exposed because of their occupation. And for immunocompromised individuals, systemic infections such as bacteremia and meningitis may occur. All right. Again, For extraintestinal infections with Plesiomonas shigelloides, this could either be because of occupational exposure or if the person is immunocompromised. Plesiomonas shigelloides will not grow on TCBS, all right? TCBS, or the thiosulfate citrate bile salt sucrose agar, is the culture media typically used for the selective and differential isolation of Vibrio species. Okay, so ulitin natin, hindi po tutubo ang Plesiomonas shigelloides dun sa culture media na ginagamit for Vibrio. Eosin methylene blue and McConkey agar may exhibit the growth of some strains, and any other culture media is fine, but the inositol brilliant green bile agar is the most recommended for the isolation of Plesiomonas shigelloides, and the colonies will appear shiny, opaque, with a slightly raised center and a white to pink pigmentation, as we can see right here on this photo. All right, so again, the best culture media for the isolation of Plesiomonas shigelloides is the inositol brilliant green bile agar. The biochemical test reactions of Plesiomonas shigelloides will help us in differentiating it from Aeromonas versus Vibrio versus all the other members of Enterobacterales. So while all the other members of Enterobacterales are oxidase negative, Plesiomonas shigelloides is oxidase positive. Next, Aeromonas is resistant to the O129 vibriostatic agent, but Plesiomonas shigelloides is also susceptible to the O129 the vibriostatic agent. All right, ulitin po natin, katulad ng ibang mga Vibrio species, sensitive din po ang Plesiomonas shigelloides sa O129. Now, both Vibrio and Aeromonas cannot ferment inositol, but Plesiomonas shigelloides is an inositol fermenter, which is why, as we've mentioned earlier, its preferred culture media is the inositol brilliant green bile agar. And finally, as shown here on the photo to the left, Plesiomonas shigelloides will thrive in a nutrient broth with 0% sodium chloride, but it will not grow on a nutrient broth with added 6% sodium chloride because, again, they are intolerant to the increased salt concentration. All right? Do take note, though, that the same is true for Aeromonas. Okay, ulitin po natin, kung ikukumpara natin, ang Vibrio species po kasi, they are mostly halophilic. Kaya mag-grow sila with the supplementation of 6% sodium chloride. But for Aeromonas and Plesiomonas shigelloides, they are intolerant to increased salt concentration, which is why they would grow on a nutrient broth with 0% salt, but they would not grow if there is 6% or higher concentrations of salt. All right. So Plesiomonas, to be honest, its classification seemed very confusing for some time, diba? Serologically, it could cross-agglutinate with the Shigella species. Pero biochemically, parehas naman po silang oxidase positive nung Plesiomonas at saka Aeromonas. Pero hindi sila parehas na susceptible to the vibriostatic agent. But then, further study showed that it's more closely related to Enterobacterales, 'di ba? So parang saan ba talaga belong ang Plesiomonas, 'di ba? Pick a side, no? Medyo confusing siya. But again, Plesiomonas shigelloides is now under the order Enterobacterales. May mga similarities and differences lang po siya between Vibrio species and Aeromonas species. So again, this slide summarizes the biochemical test results that's going to help us differentiate Plesiomonas shigelloides among the others. Next is the Proteus species, commonly isolated from urine, wound, and ear infections. They can also infect the proximal kidney tubules and cause acute glomerulonephritis or AGN. They're not simply just urease positive, but they have also been noted as rapid urease producers. So here's the thing. When urease breaks down urea, it produces an alkaline urine pH. Now, the normal urine pH is slightly acidic to neutral, but during Proteus infections, it raises the pH to become alkaline, which usually encourages the formation of some renal crystals, which eventually accumulates as renal stones. All right. So in your um future analysis of urine and other body fluids, so yung AUBF classes nyo po, matututunan nyo po that there are different types of urinary crystals as well as at which pH environment do they form. Okay? So some crystals, they form at an acidic pH. Yung iba naman po, they don't care. No? Acidic man or neutral, pwedeng mag-form yung urinary crystals. But these specific crystals could form when they are in an alkaline environment. Okay. And one of the reasons that contribute to the formation of those urinary crystals that eventually turn into renal stones is ito nga po, no, the alkaline environment that Proteus has created. Okay. So ulitin po natin, no, Proteus species are rapid urease producers, which is associated with renal stone formation. Okay, so ulitin natin anong kaugnayan nun. The production of urease leads to the breakdown of urea and produces an alkaline environment. So again, there are some renal crystals that are formed when the pH of the urine is alkaline. Okay? And pag pinagsama-sama po natin yung mga urinary crystals na yon, this could lead to the formation of renal stones. All right? So again, that is the association between Proteus species being rapid urease producers to renal stone formation. Kapag may urinary tract infection ang isang individual na Proteus yung may kasalanan, maaari din po itong mag-progress into renal stone formation. All right? Now, there are many other species within the Proteus genus, but only these two are considered as human pathogens. So, Proteus mirabilis and Proteus vulgaris. Take note of their characteristic colony descriptions. They produce clear, non-lactose fermenting colonies with a swarming motility on McConkey, as we can see right here on this photo. And even on BAP po, pwede rin natin ma-observe yung swarming motility nila. Minsan po, kahit nakalimutan nyo or ma-confuse kayo sa mga biochemical test results ng kahit alin sa mga bacteria na na-discuss na natin, there will always be these couple of things that would still help you recognize them. Okay? So, wag na wag nyo pong kakalimutan yung mga naka-highlight dito. Okay? So, again, Proteus species are capable of producing swarming colonies, and they could also produce a burnt chocolate or burnt gunpowder odor. Okay. Next, we could also look into what diseases are they associated to, and ano ang special colony morphology description nila and other stuff like that. All right. So again, if for example nakakalimutan niyo yung IMVIC nila or yung TSI reactions nila, you could look for other things that is unique to the organism. So again, for Proteus species, they may be associated with the renal stone formation or acute glomerulonephritis. And with regards to their colony morphology, ayan nga po, no, maraming iba't ibang organisms ang napag-usapan na natin na non-lactose fermenter, pero Proteus lang ang may capability to produce a swarming phenomenon. Here are the biochemical test reactions of both Proteus mirabilis and Proteus vulgaris. So on IMVIC, the V here means that their reactions could be variable. Ang sigurado lang po tayo dito ay yung kanilang indole reaction. So for Proteus mirabilis, its indole production is always negative, but for Proteus vulgaris, its indole production is always positive. All right. Another test that could be used to differentiate mirabilis and vulgaris is ornithine decarboxylase. All right, Proteus mirabilis would test positive, while Proteus vulgaris is negative. Naka-highlight dito ang PDA or yung phenylalanine deaminase test. Now, they're both positive. Now, what is significant about testing positive for phenylalanine deaminase? among all Enterobacterales members that we will be discussing, tatlo lang po ang nagpa-positive sa phenylalanine deaminase. Okay? So it's PMP. It's Proteus, Morganella, and Providencia. Okay? So ulitin po natin, kung halimbawa nabigyan tayo ng case study question where we are expected to identify the organism based on the biochemical test results shown, if nakita nyo po doon sa listahan na kasama yung phenylalanine deaminase at nag-positive siya sa phenylalanine deaminase, tatlo na lang agad ang choices natin. And again, it's Proteus, Morganella, and Providencia. Dahil Proteus, Morganella, and Providencia lang po ang Enterobacterales member na kayang mag-positive sa phenylalanine deaminase. Next is Morganella morganii, which may be very easily confused with Proteus vulgaris because almost all of their biochemical test results are the same. So ano po ang points of differentiation nila? Okay? Morganella morganii does not produce swarming colonies. Okay? Inuulit ko po kapag nakita nyo na Enterobacterales member siya and it produced swarming colonies, then dapat ang una nyong maisip is the Proteus species. But if all the other biochemical test results is the same, pero hindi nagpo-produce ng swarming colonies, then you're most likely looking at Morganella morganii. Okay? Also, Morganella morganii is negative for hydrogen sulfide reaction. Kabaliktaran ng Proteus species, which is positive for hydrogen sulfide production. All right? But again, among their similarities is PAD or phenylalanine deaminase dahil parehas positive for phenylalanine deaminase ang Morganella, Proteus, and Providencia. The genus Providencia is composed of 5 different species: Providencia alcalificans, Providencia stewartii, Providencia ritgeri, Providencia rustigianii, and Providencia haimbacae. Providencia ritgeri has been documented to cause diarrhea and UTI, so it's primarily a urinary tract pathogen. Providencia stewartii has been implicated in outbreaks in burn units and has been also isolated from urine cultures. And Providencia alcalificans has been seen to also cause a similar disease to the traveler's diarrhea. Okay. For Providencia rustigianii, it has unproven pathogenicity. Now, the infections caused by Providencia stewartii and Redgeri, especially in immunocompromised patients, are partific, um, particularly difficult to treat because of their resistance to antimicrobials. Listed here are their biochemical test results. So on IMVIC, they are positive, positive, negative, positive. So Voges per scour lang po ang negative for the Providencia species. Its TSI reaction is also K over A or alkaline over acid, and it's negative for both gas and hydrogen sulfide production. And again, as previously mentioned, no, Providencia species is one of the 3 Enterobacterales members capable of testing positive for PAD or phenylalanine deaminase. So ulitin po natin, what are the only 3 Enterobacterales members capable of testing positive for phenylalanine deaminase? PMP, di ba? Proteus, Morganella, and Providencia. Serratia species are now under the family Yersiniaceae. Okay, so ulitin ko po, no? Under the order Enterobacterales, andun yung family na Enterobacteriaceae. Pero dati, mag-isa niya lang doon. At lahat sila nasa loob ng family Enterobacteriaceae. Pero ulitin po natin, na-reclassify na po sila. So Serratia now belongs to the family Yersiniaceae, which would include Yersinia pestis, but we'll talk more about that later. There are 2 common isolates, Serratia marcescens and Serratia rubidea. Now, Serratia are opportunistic nosocomial pathogens that has been associated with bacteremia in nurseries, cardiac surgery units, and burn victim units. So almost anywhere in the hospital, really. Some strains are late lactose fermenters, which is determined as they test positive for ONPG, or the ortho nitrophenyl beta D galactopyranoside test. Okay, so again, the ONPG test is done to determine if the organism is a late lactose fermenter. Kung wala ka ng oras para i-extend yung incubation to further observe if this non-lactose fermenting organism eventually is capable of fermenting lactose, you may perform ONPG. So again, through the ONPG test, we are capable of determining if an organism is a late lactose fermenter. Okay. On McConkey, we may be able to observe its characteristic brick red colonies due to the production of the pigment prodigiosin, specifically when incubated at 25 degrees Celsius. Okay, ulitin po natin. On the McConkey agar, Serratia species are clear and non-lactose fermenting within the 24-hour incubation period. But if you incubate your McConkey agar at 25 degrees Celsius at Serratia yung isolate ninyo, you would be able to observe its brick red colonies. And again, the pigment responsible for these brick red colonies is proteasin. So listed here are its IMVIC and TSI reactions. So ang IMVIC po ng Serratia is negative, negative, positive, positive. All right? Anong organism ang kahawig niya? Klebsiella pneumoniae, di ba? Klebsiella pneumoniae is also negative, negative, positive, positive. All right? Serratia species will also test positive for ONPG, as previously mentioned. It's also positive for gelatinase, urease, and DNase. That was for the opportunistic members of Enterobacterales. Let's now discuss the primary intestinal pathogens, or yung true pathogens ng Enterobacterales. So these ones are also referred to as the overt pathogens or true pathogens. These ones are not part of the normal flora of the human gastrointestinal tract and not anywhere else. Okay? Kahit saan, hindi po part ng normal flora itong tatlong to. And being able to isolate them from patient samples is considered as a very significant clinical finding. So there are three bacterial genera recognized as primary intestinal pathogens: Salmonella, Shigella, and Yersinia. Members of the genus Salmonella produce significant infections in humans and in certain animals. Many Salmonella serotypes are typically found in cold blooded animals, as well as rodents and birds, which serve as their natural host. Now, according to Mahon, there was this time in America where a lot of people were documented to have acquired salmonellosis from their pet frogs. um you could check on that. You could read your Mahon. That's true. Um, Having a frog as a pet is a questionable decision. So kung nagkaroon ka ng salmonellosis from having a pet frog, I say deserve. Just kidding. Um, Salmonella species in general are gram-negative, facultatively anaerobic bacilli that morphologically resemble any other enteric bacteria. So straight gram-negative rods din po sila. Infections may be acquired either from ingesting contaminated and improperly cooked food or direct contact with human carriers. Right? So factors responsible for the virulence of Salmonella would include the fimbriae used in adherence and initiating intestinal infection. Another um, Another virulence factor is enterotoxin production. Okay? So, enterotoxin produced by some Salmonella strains could cause gastroenteritis and has been implicated as a significant virulence factor. Lastly, Salmonella species also have the capability to move across intestinal mucosa. Salmonella species have antigens similar to those of other enterobacteria, and meron din po sila somatic O antigens and flagellar H antigens, and These would serve as their primary antigenic structures used in serologic grouping of Salmonella. So para naman po sa mga Salmonella, their K or capsular antigen is otherwise known as the V or VI antigen. Okay, the serologic identification of the V antigen is important in identifying Salmonella serotype typhi. Okay, currently, based on their DNA homology and sequencing, the genus Salmonella is composed only of two species. So, first is Salmonella enterica, and under that is six subspecies. So, we have Salmonella enterica subspecies enterica, subspecies salame, subspecies arizone, subspecies diarizone, subspecies Hortene, and subspecies Indica. Now, within the Salmonella enterica subspecies enterica, there are about three serotypes. All right, so we have serotype typhi, serotype coloraceus, and serotype paratyphi. Next species in this genus is Salmonella bongori, which is rarely isolated and was named after the town of Bongor in Africa. And this species is initially isolated from a lizard, and that was initially found among cold-blooded animals. Okay, so ulitin po natin. Salmonella bongori was first isolated from a lizard. 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 There are two categories of infection for Salmonella enterica. First is gastroenteritis, commonly termed as food poisoning. So, listed here are its modes of transmission and clinical manifestations. All right. So, through poultry products, contaminated kitchen utensils, and pet handling. So, gastroenteritis or food poisoning caused by Salmonella enterica leads to vomiting and diarrhea. Next is enteric fever or the typhoid fever, caused specifically by Salmonella enterica, subspecies enterica, serotype typhi, or simply Salmonella typhi. This is a febrile disease that can be acquired through any of the following ways listed here. So, ingestion of food prepared by infected individuals or carriers, ingestion of contaminated food and water, or direct transmission through fomites. So dito rin po nanggaling yung story about Typhoid Mary. Hindi ko na po ikakwento yung buong thing. I-search nyo na lang, no, Typhoid Mary. But there was basically this cook na nagluto siya ng pagkain for a lot of people, but carrier pala siya ng Salmonella typhi. So everybody else who ate her food eventually acquired enteric fever or typhoid fever. All right. So that's why she was known as Typhoid Mary. All right. So clinical manifestations include anorexia, myalgia, severe frontal headache, and the appearance of what has been termed as rose spots, which may be seen anywhere on the patient's body and is usually an indication of untreated typhoid fever. So, as shown here on this picture, no, this is how rose spots would look like up close. So, again, that is the hallmark of infection for enteric fever or typhoid fever. Salmonella enterica are hydrogen sulfide or H2S producers. But on McConkey agar, since wala pong H2S indicator ang McConkey agar, we only see them as clear non-lactose fermenting colonies. But on SSA or the Salmonella Shigella agar, which we'll also be talking about later, dito po natin makikita yung characteristic fish eye colonies nila described as clear colonies with black centers. Now, those black centers is because of their ability to produce hydrogen sulfide. And here are its biochemical test results. On IMPIC, it is negative, positive, negative, negative. Okay, so ang ipapositive lang po ng Salmonella enterica is yung methyl red test. All right. Next on TSI, it is K over A or alkaline over acid. Negative for gas production, but positive for hydrogen sulfide production. Next is the Shigella species, and this genus is actually very closely related to Escherichia. Kaya po, diba, if you would remember from our discussion earlier on the different strains of E. coli, EIEC has been described to produce a Shigella-like infection because it's able to invade intestinal cells. Meanwhile, yung EHEC naman po, on the other hand, it produces a Shigella-like toxin. Now, on McConkie agar, their colonies look just like any other clear non-lactose fermenting colonies. But on SSA, or the Salmonella Shigella agar, they kind of look similar to Salmonella except that they don't have black centers because negative for hydrogen sulfide production po ang Shigella species. Now, as we can see right here on this table, there are four different species classified into four different serogroups. So the basis for their serogrouping would be their somatic O antigen found on their cell walls. Okay, so Shigella dysentery is a group A, Shigella flexneri is group B, and Shigella boydii is group C, and then Shigella sonei is group D. So just try your best no, to not get them mixed up or try to make a mnemonic for this. So nung time po namin, no, yung MTAP prof namin, ang mnemonic na binigay niya is, don't F boys. So in that order, no, D is dysentery, which is group A. F is flexneri, which is group B. Boy is boy DI, which is group C, and S is group D. So don't F boys. Bahala ka na kung anong ibig sabihin ng F, 'di ba? Ako, I'm a family-friendly channel. So F might mean fight, don't fight boys, 'di ba? But again, that's for the different serial groups of Shigella species. Let's first look at the general biochemical characteristics that is applicable to all the four species, plus some exceptions. So first, all Shigella species are non-motile. All right. Now, if you would remember from our discussion on the general characteristics, diba? Ano po yung mga non-motile Enterobacterales members? SKY, diba? Sky, Shigella, Klebsiella, and Yersinia. Next, all Shigella species are negative for urea hydrolysis, hydrogen sulfide production, and lysine decarboxylase. Okay, lahat ng Shigella, lahat ng apat na Shigella species, they are negative for urea hydrolysis, hydrogen sulfide production, and lysine decarboxylase. Next, all Shigella species are mannitol fermenters except for Shigella dysenteriae. All Shigella species are negative for gas production except Shigella flexneri, and all species are ornithine decarboxylase negative except for Shigella sonei. So again, here are their um, general biochemical characteristic with some exceptions. Now, on the second column, there are other specific characteristics per species. So first up is Shigella dysenteriae. This is the most virulent out of all of them and is mainly responsible for causing bacillary dysentery. Okay, so ito rin po yung nagpo-produce ng Shiga toxin, and here is a list of their biochemical test results. Next, in the past naman po, Shigella flexneri has been noted as one of the causes of gay bowel syndrome, along with Giardia intestinalis or Giardia lamblia. Okay, so ayun po no. Again, in the past, no, in the past, historically, no, there has been this disease that has been associated with homosexual persons. So again, that was referred to as the gay bowel syndrome. And yung two organisms po associated with the development of the gay bowel syndrome was Shigella flexneri and Giardia intestinalis. All right. So ayun po. But again, no, hindi na ginagamit yung term na gay bowel syndrome because we have established na hindi lang naman homosexual interactions yung mode of transmission ng ganitong diseases. Alright? So that was for Shigella flexneri. Next is Shigella sonei. So Shigella sonei only causes a self-limiting form of watery diarrhea. Shigella sine is also a late lactose fermenter, so it means that it would test positive for ONPG. Let's now discuss bacillary dysentery, which is caused specifically by Shigella dysenteriae type 1. Here are its modes of transmission, and its clinical manifestations include nonspecific symptoms such as fever, chills, abdominal cramps, painful bowel movement, and this may also cause rectal prolapse in children due to the excessive straining of the rectum. So just in case you're not familiar with this, no, um rectal prolapse is a condition that may have also been discussed in your parasitology classes. Trichuris trichuria has also been documented to cause rectal prolapse among children, which happens when a portion of the rectum protrudes to the outside of the anus, turning itself inside out, as we can see right here in this illustration. The more serious clinical manifestations of bacillary dysentery include acute inflammatory colitis and bloody diarrhea that is composed of not just blood but also WBCs and mucus. So one of the complications of bacillary dysentery is hemolytic uremic syndrome. Ulitin po natin, cases of bacillary dysentery ay maaaring tumuloy into HUS. Next is Yersinia pestis. This is commonly known as the plague bacillus and is the causative agent of the bubonic plague or the Black Death that caused the death of approximately 75 million to 200 million people across Europe, Asia, and many other regions during the 14th century. Yersinia pestis has been classified as a type A bioterrorism agent by the Centers for Disease Control and Prevention. Recall time? Recall time muna po. There were two other bacterial pathogens na na-discuss na natin in the past na category A bioterrorism agents then. So one was the aerobic spore-forming gram-positive bacilli, and the other was a gram-negative fastidious organism. So, do you know, no? If you know, i-comment nyo below. No? If you know, comment below. Ano po yung dalawa pang category A agents of bioterrorism ang na-discuss natin in the past? First is Bacillus anthracis, di ba po? And the next one is Francisella tularensis. Okay, so ulitin po natin, if the organism has been described to be aerobic, spore-forming, and gram-positive, ang pinag-uusapan ay Bacillus anthracis. Pero kung gram-negative fastidious organism, ang pinag-uusapan ay Francisella tularensis. But in this case, no, kapag po straight gram-negative bacilli siya with a safety pin appearance, ang pinag-uusapan ay Yersinia pestis. Ulitin natin, ano ang common sa tatlong yon? Silang tatlo ay Category A, bioterrorism agent by the Centers for Disease Control and Prevention. All right, again, the three organisms that we have already discussed that are classified as Category A bioterrorism agents include Bacillus anthracis, Francisella tularensis, and Yersinia pestis. Okay, so ituloy po natin ang discussion on Yersinia pestis. Nabanggit din kanina, di ba, sa general characteristics that all of them are motile except for Sky, SKY, diba? Shigella, Klebsiella, and Yersinia. So again, Yersinia is non-motile. Virulence factors of Yersinia pestis include coagulase and fibrinolysin. Now, una po nating natutunan ang mechanisms of action and function ng dalawang yon when we were learning about the virulence factors of Staphylococcus aureus, diba? So again, coagulase and fibrinolysin, similar to the coagulase and fibrinolysin produced by Staphylococcus aureus. Medyo naiiba po yung microscopic morphology ng Yersinia pestis compared to all the other Enterobacterales members because although yes, it is still considered as a straight gram-negative bacilli with rounded ends, it has an odd staining pattern that has been described as bipolar or safety pin in appearance, as we can see right here in this picture. So this is because they have inclusion bodies. Okay? And yung inclusion bodies po na yun, mas nagko-concentrate or mas naiipon doon yung gram stain, giving it a darker appearance. And again, this dark appearance of the poles of the bacteria has been described to be safety pointy pin in appearance. All right, so that's for Yersinia pestis. Here is its list of different culture media as well as its biochemical test reactions. So on BAP, they produce pinpoint colonies. On MAC, they are clear non-lactose fermenting. And if you use a broth culture, Yersinia pestis is capable of producing a stalactite pattern. On IMVIC, its IMVIC result is negative, positive, negative, negative. So, methyl red test lang po ang kayang i-positive ng Yersinia pestis. On TSI, it is K over A, so alkaline over acid, and it's negative for both gas and hydrogen sulfide production. There are two forms of the plague caused by Yersinia pestis. First is the more popular bubonic plague, and unlike all the other members of Enterobacteriaceae, ang mode of transmission po nito is not via ingestion, but rather it is acquired from being bitten by an infected flea belonging to the species Synopsilla tropis. So this photo shows us the clinical manifestations, and if you would direct your attention to the bottom right image, kaya po ito tinawag na Black Death because it causes necrosis of the tissues, turning it black once the infection has reached the septicemic stage. Okay, ulitin po natin. Sa bubonic plague or black plague, nagkakaroon ng peripheral necrosis yung pasyente kung saan nagkakaroon po ng blackening or necrosis dun sa periphery ng pasyente. Okay, so aside from that, there's also high fever and the formation of buboes. All right, so buboes is the swelling of lymph nodes. Next is pulmonary plague. Dito naman po, this only occurs secondary to the bubonic plague. And kahit walang flea vector, people could acquire this through close contact with other infected individuals. Yersinia enterocolitica is the most commonly isolated species of Yersinia, and it causes infections typical of enterobacteria such as waterborne gastroenteritis or enterocolitis. Do take note, though, that this pathogen has the ability to cause an infection whose clinical manifestations can mimic those of someone who is suffering from appendicitis, which is why we have the term appendicitis-like syndrome. Okay, ulitin po natin. Yersinia enterocolitica is capable of causing an infection that has been described as an appendicitis-like syndrome. So yung clinical manifestations daw ay similar with a person suffering from appendicitis. Now, we've established the fact that Yersinia in general are non-motile, but Yersinia enterocolitica is different. All right. So at 22 degrees Celsius, it could give a motile result on SIM. Again, what does SIM stand for? It's the sulfide indole motility medium. All right, so on SIM or the sulfide indole motility medium, maaari daw po na mag-produce ng positive for motility ang Yersinia enterocolitica, specifically if you incubate it at 22 degrees Celsius. But we won't be able to observe this at the usual 35 to 37 degrees Celsius. So basically, kikilos lang siya kapag medyo malamig yung environment. All right. So, one selective media that can be used to isolate Yersinia is the Cephsulidin Irgasan Novobiasin agar or CIN. So on CIN, Yersinia enterocolitica will produce transparent colonies with dark red centers, being described as the bull's eye colonies, as we can see right here in this photo. So on IMVIC, its indole production would vary, but we are sure that this one is MR positive, VP negative, and citrate negative. All right. So again, the IMVIC result of your enterocolitica is variable, positive, negative, negative. On this table, we could see the different TSI and LIA reactions that the different members of Enterobacterales can produce. The problem here is that some organisms fall into two or more different interpretation. So, tignan po natin, for example, yung Proteus vulgaris and Proteus mirabilis, which have been noted to produce A over A and positive for H2S reactions. But if we look right over here, okay, nakikita po natin dito that it's also capable of producing K over A and positive for gas and H2S positive result. Alright? Next naman po, hanapin natin kung nasaan ang E. coli. Alright? So dito po, we're capable of seeing that E. coli, no, has been known to be A over A, positive for gas, negative for H2S. But there are strains of E. coli that is K over A, alkaline over acid, and then positive for gas, diba? And also some of them is K over A and also negative for gas production. Okay, so nakikita natin that one organism may be able to produce multiple different reactions on PSI or LIA. So sometimes it may be very confusing. So kailangan po makilala talaga natin yung bawat organism, not just based on these test results, but also based on their own unique characteristics, such as what diseases do they cause, what do they look like on different culture media, and other stuff like that. Para matulungan po tayo na ma-identify itong mga organisms na to, if, for example, nalilito na tayo sa mga biochemical test results nila. Let us now look at some of the selective and differential culture media that we could use to isolate these Enterobacterales members. The McConkey agar, or MAC, was the first solid differential media to be formulated, which was developed in the 20th century by Alfred Theodore McConkey. So, ang pangalan po ng McConkey agar ay nanggaling sa pangalan ng person na nag-develop nito. So, the McConkey agar is a selective and differential media used for the isolation and differentiation of non-fastidious gram-negative rods, particularly members of the family Enterobacteriaceae. This differentiation is mainly based on the bacteria's capability to ferment lactose or not. Okay, ulitin po natin. Ang primary characteristic na gusto nating tignan gamit ang McConkey agar ay lactose fermentation. Okay? So, listed here are the key components of the McConkey agar. Its only carbohydrate source is lactose, specifically lactose monohydrate. So wala na pong ibang sugar content ang McConkey maliban sa lactose. Its inhibitory agents include bile salts and crystal violet. So the purpose of bile salts and crystal violet is to inhibit the growth of most gram-positive bacteria and fastidious gram-negative bacteria. Uulitin ko po, bile salts plus crystal violet will prevent the growth of almost all gram-positive bacteria and fastidious gram-negative bacteria. So, yun po yung dahilan kung bakit siya naging selective. Okay? So, if we're inhibiting the growth of gram-positive bacteria, ano na lang ang tutubo dito? Using the McConkey agar, we would be able to grow non-fastidious gram-negative bacteria. So here's what's going to happen. If the bacteria is able to ferment lactose, fermentation leads to the production of acids, di ba po? And this acidic environment would now react with the neutral red. All right? Ang pH indicator po ng McConkey agar ay neutral red. So neutral red is the pH indicator, at kapag sinabi po nating pH indicator, this component will show us if there is a change in pH within the culture media. So now, the acids that were produced by the fermentation of lactose will react with neutral red, producing colonies that are pink. Okay, so again, lactose fermenting bacteria will appear pink using the McConkey agar. But if, for example, the bacteria did not utilize lactose in the culture media, then no acids will be produced. So if there is no acids produced, nothing will react with the pH indicator. And this will lead to non-lactose fermenting bacteria that are clear or colorless. Okay, so ulitin po natin, lactose fermenting, pink colonies. Non-lactose fermenting, colorless colonies. All right, so that's for the McConkey agar. This slide shows as an example of a lactose fermenting bacteria that produced pink colonies, as we can see on figure A. And in figure B, this is what it looks like if the bacteria is non-lactose fermenting. The McConkey sorbitol agar was formulated by removing lactose and replacing it with sorbitol into the original McConkey formula for us to be able to differentiate EHEC 0157H7 isolates from all the other E. coli strains. Okay, ulitin po natin. Ang purpose ng McConkey sorbitol agar is to differentiate the E. coli 0157H7 in comparison to all the other E. coli strains. So, um, Escherichia coli 0157H7 is negative for sorbitol fermentation. Okay, ulitin po natin. E. coli 0157H7 is not capable of fermenting or utilizing sorbitol. Meanwhile, all the other E. coli is positive for sorbitol fermentation. So, ang mangyayari po dito, same principle no with lactose fermentation. Once sorbitol has been fermented, it would produce acidic byproducts. The acidic byproducts would react with the neutral red. So, if the organism was able to utilize sorbitol, it would produce pink colonies, all right? But if your isolate is E. coli 0157H7, it's not capable of utilizing sorbitol, so it would produce clear or colorless colonies, as we can see right here on figure B. Okay, so again, the purpose of the McConkey sorbitol agar is to differentiate Escherichia coli 0157H7 from all the other Escherichia coli strains. The Eosin methylene blue agar, or in some references is referred to as the Levine's medium, is used to differentiate lactose fermenting colonies such as E. coli that produces its iconic green metallic sheen. Now, if the bacteria is also lactose fermenting, but it's not Escherichia coli, then their colonies would appear to be purple, like this one. Now, this one right here is an example of Enterobacter aerogenes or Klebsiella aerogenes on EMB. Non-lactose fermenting Enterobacterales members will appear colorless, and other coliforms that belong outside of the family Enterobacteriaceae or order Enterobacterales will grow pink colored colonies, as we can see right here. So ulitin po natin, no, the eosin methylene blue agar or EMB, its pH indicator is eosin and methylene blue. All right. Now, if the organism is a lactose fermenter, there are two ways on how this could go. If it's Escherichia coli, then it would produce the iconic green metallic sheen. But if it is anything other than Escherichia coli na member din naman ng Enterobacterales, we would be able to observe purple colored colonies. If the organism is non-lactose fermenter, colorless. But if it is a lactose fermenter, but it's not a member of Enterobacterales, then it would produce pink colored colonies. All right. So basically, the eosin methylene blue agar is a selective and differential medium. Next is the hectone enteric agar, composed of bile salts and bromothymol blue. That's supposed to inhibit the growth of other gram-negative bacilli for better isolation of Salmonella and Shigella. All right, ulitin po natin, no? The hectone enteric agar promotes the isolation of Salmonella and Shigella, and it has, ayan po, bile salts and bromothymol blue to inhibit the growth of other gram-negative bacteria. But at the same time, pH indicator niya rin po ang bromothymol blue. It also has H2S indicators capable of showing us hydrogen sulfide production. And its H2S indicator is ferric ammonium citrate. All right. So lactose fermenting Enterobacterales members would be able to produce yellow colored colonies. Meanwhile, non-lactose fermenting organisms would produce blue-green colonies. Tapos, depende na lang po yun if they produce hydrogen sulfide. If they do, then they would also have black centers. Meanwhile, non-enteric pathogens will produce orange or pink orange colonies such as this one right here. Okay, so those are the different colony morphologies that we may be able to observe using the hectone enteric agar. But again, this is mainly to promote the isolation of Salmonella and Shigella. Next is SSA, or the Salmonella Shigella agar, which is specifically formulated to differentiate Salmonella and Shigella species from other enteric bacteria. So its components would include bile salts in brilliant green, which is supposed to inhibit the growth of gram-positive organisms. Next, its pH indicator is neutral red. So kanina po napaulit-ulit na nating na-explain what the purpose of a pH indicator would be, but it also has a hydrogen sulfide indicator, which is sodium thiosulfate and ferric ammonium citrate. So it's going to show us if the organism produces hydrogen sulfide. And as for its carbohydrate source, it contains lactose. So Shigella species would be able to produce colorless colonies. All right. Next, for Salmonella, it also produces colorless colonies but with black centers dahil ulitin natin, non-fermenter ito ng lactose but it produces hydrogen sulfide. So again, these are referred to as the fish eye colonies of Salmonella. Last on our list is the xylose lysine deoxycholate agar. And ang pinakakailangan lang po natin tandaan dito is that this is used for the isolation of Salmonella and Shigella from heavily contaminated samples such as stool. So its pH indicator is phenol red. It also has hydrogen sulfide indicators, and how to interpret the growth in XLD. First, if it was able to produce pink or red colonies with no black centers, it's Shigella. If it produces pink or red colonies with black centers, then it's Salmonella. And again, saan galing yung black centers? It's because of the hydrogen sulfide production. And next, for all the other Enterobacterales members, they're capable of producing yellow colored colonies. In this example, what we have right here is Escherichia coli. Here are the references that were used in the making of this discussion. That ends our discussion on Chapter 19 of Mahon's textbook of Diagnostic Microbiology, which focused on the order Enterobacterales. This has been Aurel Enriquez. Thank you so much for watching.